Well, hi everybody. Welcome to iGEM Concordia's mini Jamboree. So for the next couple of days, we're going to be having some workshops, talking to some speakers. It's going to be a lot of fun. A lot of interesting people are going to be here. Speaking of interesting people, we have our first speaker, Luchino Cohen. So Dr. Cohen is currently employed at the Canadian Space Agency as a senior exploration scientist within the Astronauts, Life Science, and Space Medicine group, which is probably one of the coolest titles I've heard of in a long time. So I'll let him get started, and thank you so much for coming.
So good morning, everyone. I'm very happy to participate in this great event and talk to you about our perspective in regarding synthetic biology at the Canadian Space Agency. Just a little bit of background. I'm a member of a group called Health and Life Science, and we are interested in supporting research, external research, in order to mitigate health risks of spaceflight for humans. And for that, we use various platforms, such as ground laboratories. We're supporting research on the International Space Station. And we are also interested in using microgravity analogs, which are models that can reproduce microgravity effects, such as, for example, bed rest studies. And so we have been interested, especially starting last year, about synthetic biology, because there are interesting potential space applications of synthetic biology. In particular, we learned that there was a possibility to use cell-free systems for producing bioactive molecules. That would be a way to produce these molecules in space during space missions, or to actually use these cell-free systems to perform point-of-care medicine. The possibility to modify these microorganisms to produce probiotics that would produce nutrients, antioxidants, or biological active molecules. There was also some applications such as using cell-free systems for performing medical diagnostics for viral and bacterial infections. So these microorganisms could be engineered to produce, for example, radioprotective molecules that could be then administered to the astronauts to reduce the effect of radiation exposure during spaceflight, or to produce a supplement like vitamins and other nutrients to help them cope with the harsh conditions of spaceflight. Now, on Earth, of course, you all know that this microorganism can be modified to correct food intolerance or metabolic impairment. So the Earth applications are very important for us, and we are trying to find solutions that fit both space problems as well as Earth problems. Some examples of molecules that could maybe provide radioprotective effects are illustrated here. If there's any way to produce these molecules through genetic engineering, that could be a potential application for space missions. Now, several space agencies have already started supporting research in synthetic biology. And you can maybe see here some examples of projects that were supported by NASA, by Roscosmos, the Russian space agency, as well as by the Chinese space agency. And in blue, there are a few examples of projects that are performed on the ground by NASA and supported by NASA. So all of these projects are using usually microorganisms, but some of them are using plants. It's just a start where all the agencies are getting interested in this field and see if there's any potential solutions to specific space mission issues. And when I'm talking about space mission issues, we are all geared towards going to the moon and then to Mars. And the NASA has published since a long time a path into risk reduction, to health risk reductions. And this is a table published by the Human Research Program at NASA. They update it regularly, and you can see that some of these issues, such as radiation exposure, Reynolds storm formation, medication long-term storage, are still there's no definitive solution, especially when we consider deep space missions like missions to Mars. And we have the feeling that the synthetic biology could contribute to solve specific issues such as radiation exposure, inadequate food and nutrition, medication long-term storage, as well as, for example, altered immune responses that has been observed during short-term as well as long-term missions, like 6-month missions in space. Actually, these issues happen even on in low Earth orbit on the ISS. But they are, of course, a major risk when we go farther from the Earth orbit. Now, NASA has started investigating the potential of synthetic biology through the collaboration with the Translational Research Institute for Space Health. This is called TRISH, and it's a virtual institute that is funded by NASA, by the NASA Human Research Program. They tackle the specific challenges of human deep space exploration. They usually fund very innovative research through breakthrough approaches. And some of these projects supported by TRISH are listed here, such as just-in-time medication that could be produced by microbial systems, in situ expression analysis of therapeutic microbes with gastrointestinal devices. You can see that this is really innovative approaches that are being tested on the ground as we speak. NASA has also NASA Ames Research Institute, which is located in California, has also a synthetic biology program. They have a SynBioApps project, and they have started developing technology to biomanufacture products on demand, such as vitamins and medicines. And you can see here an example of the bionutrient experiment that is actually happening on the ISS. And actually, our Canadian astronaut, David Saint-Jacques, participated in this investigation. They are testing an in-space nutrient production method. And so using Baker's yeast, and they aim to produce specific antioxidants such as beta-carotene and zeaxanthin. Now, so basically, several agencies are seriously considering using synthetic biology to solve problems or challenges of deep space missions. And so we started to get interested in this field and started looking at the capacity in Canada. There is a Canadian community that is interested in synthetic biology, and these are just a few examples of some groups that are working either in British Columbia, in Alberta, in Ontario, and in Quebec. And it's certainly not a complete list of all the investigators and laboratories and institutions that could contribute to this field, but we feel that there is certainly a significant community that have expertise and that may be interested in actually finding solution for space applications. And of course, the iGEM team, Concordia, is a great example of a team that may provide us with innovative solutions. Now, we also know that several funding agencies have funded projects in this field. And although there's, I don't think there's a coordinated effort on that. But if there is one, I think the CSA, the Canadian Space Agency, may be part of it in the future. And of course, we heard about the iGEM competition, the International Genetically Engineered Machine competition, which provide a potential source of innovation for future space projects. It's also a source of collaboration with external partner, and it's also for us. It could be a tool to build capacity in research project related to spaceflight. So last year we tried to get a better understanding of how many teams were involved in this competition, and we were really impressed by the number of teams from Canadian University that participated in this competition. And so that's another aspect of synthetic biology that is of great interest for us. And so basically that concludes my overview of where we are. We are just still in the exploration phase, but some of us think that there's a great potential for synthetic biology to solve health issues in space exploration. And we look forward to get more involvement in this field of research in the future. Thanks for your attention, and I hope you have a great day.
Thank you so much, Luchino. That was super interesting. And I'm just gonna see if there's any questions that come in the chat. Not. So, sorry, I was trying to think of a question.
So I'll be staying for the part of the day, so I'll be happy to answer questions on the chat.
Well, actually, there's one question in the chat, and it's very interesting. So can you speak more about the cell-free systems that you mentioned earlier on?
Sure. So there are teams that have investigated the possibility to use cell-free systems to produce medical, basically drugs or nutrients. I think the idea behind this type of research is to use specific microorganisms. I don't have exactly the name of the microorganisms that were used in this example, but the idea is that this solves one of the issues, that is preserving preservation, actually shelf life of this system. And if the bacteria has to be frozen, it may require a lot of resources to keep all these bacteria or yeasts or other microorganisms in a fridge. Whereas if it's a cell-free system, there's no need to refrigerate these, and then it may be easier to preserve these enzymatic components, basically, over a longer term. The shelf life is a major concern for space missions.
Great. Oh, yeah, that's a really interesting way of looking at it, where you just take the biological aspect out of it, and then it's just, here's just the chemical machinery necessary for that stuff. Yeah, that's very interesting. So we have another question. So it's more about the CSA. And it's what are some of the current biological projects CSA is involved in, what facilities exist, and how can students reach out for internship opportunities?
Okay. And I'll answer first about the CSA. We have our headquarters is in Longueuil, in South Shore of Montreal. And we also have some satellite testing facilities in Ottawa. Basically, the headquarters is near Montreal, and we certainly accept internship applications, usually through the co-op program or through the FSWEP program, which is a program to work to get work experience in the government. And you need to work through the universities to get it to be registered in these programs. And when there's an opportunity from CSA, then you will have the possibility to apply. In terms of biological projects supported by CSA, we are mostly currently supporting human research on astronauts. So typically these are research that focus on impacts of spaceflight on bone or muscle degradation, cardiovascular deconditioning, as well as there's some vision impairment syndrome that has been identified more recently due to microgravity conditions. So we are mostly supporting physiology investigation as well as psychology research on how astronauts cope with the living conditions in a space vehicle, or basically a tin can. That's the type of research we're supporting, but we are trying to switch to more to other models like animal models, but also microbial models. And we are going to publish new announcement of opportunities for non-human models, basically.
Oh, that's really interesting, especially the broadness of what the CSA is working on. Is actually, that impressed me a lot. I didn't know there was a whole psychological aspect to it, but now to think about it, that's very important. And we have one more question. So this will probably be the last question. So one of our iGEM members was asking, so through a lot of our interviews, a lot of people were talking about cellular agriculture. And they're asking if you feel like that would be a viable solution for food production in space.
Yeah, that's a good question. We actually recently launched a new group that is focusing, actually it's not part of the health and life science, it's part of the space exploration branch. And we have a group that is actually focusing on finding solutions for food production in space. So you probably heard about the effort of NASA to grow vegetables on the International Space Station. You've all seen, of course, the movie Mars, The Martian, and we hope that things are going to happen better in real life. So that's why we are preparing for alternative solutions for food production. To my knowledge, the current approach is mostly through greenhouses, but I think there's a lot of potential for, as you mentioned, cellular agriculture, or using synthetic biology to produce nutrients. And one of my colleagues is especially interested in synthetic biology and as a potential approach.
Great. Thank you. Thank you so much for your time and for your knowledge and everything. We'd love to have you back if we have any other events, if that were working for you. So I would like to thank you again on behalf of the iGEM team. Thank you.
Thank you for the invitation, and looking forward to continue working with you guys.
We look forward to working with you too. All right. So I'm just going to kind of give everyone a quick introduction to the day and how that's going to work. So hi everyone, I'm Gabriel. I'm going to be the moderator for this whole event, so for today and tomorrow. And I am a biochemistry student at Concordia University and one of the genetic leads on our project Astro Yeast. So I'm just going to go through some quick rules and stuff for the day. So overall, keep the behavior respectful. I feel like that shouldn't be an issue for anybody here, but it's just important to mention. And so does anybody have any questions, just put in the chat. I'll go through the questions and I'll ask them to the speakers. And please keep your cameras off and remain muted for the for the duration of the event. And yeah, and overall, I would just love to thank SynBioApps, the NSERC-Create program, Concordia University, the Concordia Student Union, Promega, Dr. Ketchru, our PI, and Orly Weinberg, along with our mentors for this, helping us along the way and helping us with everything we've done so far. So thank you so much.